Concentrate and give a few examples of what this sort of research is about. So it's often been said that we know a lot more about the surface of the moon than we do about the bottom of the ocean, and soon it's likely we'll probably know a lot more about the surface of Mars than we do about the oceans as well. But the oceans are incredibly important. They do so much for our lives. We're alive because of the oceans. Every second breath you take is thanks to the oceans. 50% of the oxygen in the atmosphere comes from photosynthesis in plants in the oceans. Also the water, obviously it's the biggest source of water for every living organism on the planet. And it's a massive carbon sink. So with all the CO2 that is in the atmosphere, about 33% of what is released by human activities is absorbed by the ocean. And thanks to the ocean, we have seen a slowing rate of temperature rise. So if the oceans weren't there, temperature would be rising even faster. And they also harbour the majority of life. They cover so much of the planet, 71%, but the volume they take up, they actually contain 99% of the living space of our planet. They also influence weather and climate. They control the chemistry of our planet. So they're incredibly important, but we know so little about them. The concentrating topic of today's seminar is about climate change. So I'll just give you a quick background about what is actually changing and how is it linked to oceans. So the first one I'm sure you've all heard about are greenhouse gases, and the biggest one, especially in the media at the moment, is carbon dioxide. It's released by things like burning of fossil fuels, driving your car, all those sorts of things. And if we map over the last 2,000 years and just see down there on the x-axis of this figure, if we go back 2,000 years, we can see what the CO2 concentration has been doing simply by looking at the small air particles trapped in the Antarctic sea ice. So we can easily get a very precise measure of what it's been doing. And we can see just in the last couple of hundred years, there's been a massive spike. So we're seeing an increase in greenhouse gases. The other one is temperature. On this figure, we're going a lot further back in time. We're going back 450,000 years now. And we can see that in red we have temperature going up and down, and this sort of a cycle that's within limits. And following this very similar cycle is carbon dioxide. And it's also... Sorry, do you want me to use the microphone?
Yeah.
Alright, is that better? Maybe? Okay, so the carbon dioxide is also within this very similar pattern, and it's staying within a very precise range. But until this point here, which is in very recent history, we've seen this giant spike in CO2, so it's going way up. This figure is a little bit old. Today we're at, just stay here and not move. Today we're at about 396 parts per million of CO2. The other thing that's changing is sea level as well. So we're seeing, since the beginning of the Industrial Revolution about 250 years ago, a very fast change in sea level rise. At the moment it's about 3 millimetres per year. And there's evidence for this from in situ measures of gauges, tidal gauges, but also more recently, in red here, satellite data, which has only become available obviously in recent history. So if we look at the recent warming, on this figure at the top we can see we go back to 1850, and here we are 2010, 2 years ago. Now this figure has been normalised for the period between 1951 and 1980, so that's the baseline to this figure. So you can see deviations away from the average during that time. We can see it was lower earlier than this time, and now we're beginning to see an increase. The different figures on this curve are different monitoring stations in different places around the world, but there's a consistent elevation over time. And down the bottom we can see on a geographical level differences in where most heating occurs. So the more red the colour, the more heating. We can see around in the Arctic That's where we're seeing the greatest temperature change. And recently, just last month, there was this data which came out from NASA. And when they first got the data, they thought it can't be right. There's got to be something wrong with this data. It's showing the amount of ice melting in Greenland. On the left we have July 8th, and on the right we have July 12th. So 4 days apart. In white, that's showing that there's no melting, so in white here. And in red, in the dark red, there's 3 different satellites which have picked up evidence of melting. So in 4 days, we went from having very little melting to having 97% of Greenland showing melting on its surface. So the Arctic is an area where we're beginning to see the greatest change in temperature. A bit closer to home, and we look at the Australian perspective. This data is from the Australian Government's Bureau of Meteorology, and we again see changes in temperature, deviations away from this baseline which has been chosen as 1961 to 1990. That's an arbitrary time point chosen. It's just to show how temperature changes from that time point. We can see earlier on temperatures were much lower on a decadal scale, and temperatures have begun to rise. And in this last decade, 2000 to 2010, it's been the highest it has been in recorded history. So we've seen sea temperatures becoming much higher than we have before. But what does the future hold? Really where a lot of the research is going is trying to simulate future climate change scenarios to understand the impacts we might see and what marine life is most susceptible to these changes in the future. Up the top we've got the amount of carbon dioxide we release, and at this point here, this current day, that's the history, and these are 2 very different potential scenarios depending on the decisions that our politicians make today about how they're going to use fossil fuels to produce greenhouse gases. And we can see a business as usual scenario where we continue to burn at an accelerating rate, or we cap and we begin to decrease the amount of fossil fuels we burn. And that can lead to very different changes in the amount of warming. So now on this axis we have temperature changes, and down here we have time. So up to the end of this century, depending on what we do now, we can see up to a 7-degree increase, or we might be able to cap it at a couple of degrees. Now going on to the research that I do, it's all about the oceans and it's all about climate change. There's many things that are changing in the oceans. There's rising temperature and ocean acidification, which I'll talk more about a bit later. But there's also changes in ocean currents, the sea level, and a lot of species extinctions. At the moment, it's believed that we're in a period that in the future will be called the Anthropocene, a time of mass extinction which occurred because of human activity. So previous times, like the extinction of the dinosaurs, which was a mass extinction where we saw up to 96% of ocean organisms becoming extinct.
Sorry, man. Do it. It's on the table. That's better than that. Okay, switch that off. Sorry, everybody. There you go. Can I put it?
Is that better? Yeah, that's correctly. Cool. Okay, but the 2 I'm going to be talking about today are these first 2, the changes in temperature and changes in the amount of carbon dioxide dissolving into oceans causing acidification. Most of my research can fall into these main areas, so looking at bleaching in marine cnidarians. So a cnidarian is one of the basal animals of the animal kingdom, so it includes things like corals and anemones. Also looking at marine diseases, calcification in things like coral that produce skeletons, and also work in Antarctica looking at sea ice algae. So this is a picture, an aerial photograph of a coral reef, and this is the only living structure, this is a Great Barrier Reef that can actually be seen from space. It's all been produced through, mostly by corals, but through a symbiosis within the coral organisms. So corals are animals, but inside their tissue they have single-celled algae called zooxanthellae, or a type of dinoflagellate. But it's single-celled algae, and they live inside the animal. And it's because of those algae that structures like this exist. So it's incredibly important that we understand the role of these tiny algae, because they produce something this big. So coral reefs often exist in waters where there's very low nutrients. So there's not enough to sustain life without harvesting sunlight. So the algae play that role by harvesting sunlight, producing sugars, and then giving that energy to the animal host, the coral. Now if we go underwater, this is a typical scene you might see on the Great Barrier Reef. Lots of acroporid corals and plate corals and massive boulder-like corals. If we move towards the Caribbean, you might see pictures like this, where you have huge Acropora palmata staghorn corals. You have gorgonians and all sorts of other Nigerian animals that live on a reef. And all of them contain this symbiosis, this algae which lives inside the animal. But unfortunately, they are very susceptible to changes in temperature. A small increase in temperature as small as 1 to 2 degrees can result in coral bleaching. So this is a photograph taken on Heron Island in the southern Great Barrier Reef, where an entire acropora bed has bleached. So the process of bleaching is the loss of this algae which lives inside the coral. Once it's not there, all the coral loses its colour, so all that's left is the calcium carbonate skeleton. So you get this bleaching effect. It begins to turn white. But a bleached coral isn't necessarily a dead coral. They can regain these algal symbionts if the stress of the temperature isn't sustained for too long. But if the temperature is too high for too long, eventually they die. Unfortunately, pictures like this are becoming more common on reefs around the world. And once the coral dies, it turns to rubble, and very quickly it can become overgrown by seaweeds or macroalgae. So the entire structure and function of this reef has completely changed. Now to give you a bit more of an idea on the research that we do on coral bleaching, I'll just go into a bit of the biology. So if you have a branch of coral, which that's probably about the size of your thumb, and if you zoom in and look into one of these single polyps, we can see multiple tentacles coming out from each polyp. Then if we look inside this tissue, we can see all these little green or brown dots, and they're these symbiotic algae. They're incredibly small. In the distance of 1 millimetre, you could fit 100 of these in that distance. So they're very small, but they're incredibly abundant. In 1 square centimetre of coral, you could have over 1 million of these cells, these algae. But as I said before, a small change in temperature can result in bleaching. If 1 to 2 degrees above the average, it's not very much. So they're living at their upper thermal thresholds. This was a photo which was taken on Heron Island reef flat a few years ago during a bleaching event. And what is really interesting about it is that you have this species of coral here sitting right next door to this species, but they show very different susceptibilities to this temperature anomaly. So some species are much more susceptible, others are much more tolerant. So an obvious question is to ask why. What is it about the coral or the algae that live inside these corals that makes for this large difference in response to temperature change? So it could be the animal host, or it could be the algal symbiont. And what we've begun to discover is that there's many different types of algae at a sub-species level that can vary genetically. So the genetic code is slightly different between different types of these algae that can live inside a coral. Different types of those algae then have different thermal tolerance limits. So the genetic makeup of these algae can control the vulnerability of the coral to a bleaching event. But more evidence is continuing to come out to suggest that these algae really are the limiting factor, or the weakest link in the chain of events that leads to bleaching. So it's believed these algae, there's something about them that makes them really sensitive. And it's believed that it's something to do with photosynthesis, so the harvesting of sunlight, or the splitting of water into oxygen, the release of electrons, which then goes on to form carbon and sugars. So the base of the food chain, the most simple and essential complex which sustains almost all life on the planet. So there's something about photosynthesis in these algae which makes them really sensitive. So we've been going on for many, many years to try and understand why that is. So a lot of the work we do is up on Heron Island, so that's a picture of Australia, and on the Queensland coast just over here, about 70 kilometres off Gladstone, is Heron Island. And you've probably heard a lot about Gladstone in the news lately. It's where one of the biggest coal ports is being built. A lot of shipping. So these are some of the most common species that you'd find on the reef flat on Heron Island. So you have lots of these branching corals, some of these plate-forming corals, and massive ones. So these can get into boulders maybe a metre in diameter. Now, to measure photosynthesis in these corals, so looking at the algae that lives inside the animal, we can use a tool called an imaging pan. Now, I put this up not just because it's pretty pictures, but it enables us to see how photosynthesis is working at a very fine resolution within the corals. So here we have a branch of an acroprid coral, a branching coral, and we can see across the surface of this coral there's very different responses to the conditions that we apply. So in this row here, across these three different species, we can see evidence of differences in the EQY. Now, effective quantum yield is what that stands for. I don't want to go into the detail because it'll probably bore you, but it just stands for a measure of how healthy those algae are that are living inside the coral. So it's a measure of health. Now, if we expose a coral to bleaching events and we continue to measure the health of the algae inside this animal, we can begin to see how that temperature change affects photosynthesis. Now, if we just look at this one here, we can see that it's cycling through, so it starts at control and it goes through an 8-hour time period, so it cycles through these pictures. Now, if you look at the surface of this image, we can see that it's obviously a false colour scale, and we can see a quick reduction in these values. So it starts up here in the blue-green and it quickly goes down towards the red and then black. This is telling us that within 8 hours, this branch of coral exposed to bleaching conditions has completely shut down photosynthesis. So it's very sensitive. A lot of the work we do as well is looking at marginal reefs. So Lord Howe Island, pictured down here, is the most southerly coral reef in the world. And it's really interesting, that coral reef, because it contains species which you find in other reefs around the world. But what it contains is a very unique assemblage of these symbiotic algae. Being so far south, unfortunately the corals on Lord Howe Island aren't immune from bleaching events, and we saw 2 large bleaching events both in 2010 and again in 2011. And in the lagoon area of Lord Howe, there was extensive bleaching of many corals and also anemones. What we wanted to look at was how sensitive these corals that live in this very marginal reef that are so far or so close to their limit, how sensitive are they to temperature change? And what we found was that if we increased temperature by 3 degrees, then we saw a very rapid bleaching event. So if we just saw over the seasons a change in temperature, so over years, and then we got a temperature anomaly for less than a week of a temperature outside that usual range, we saw very fast and severe bleaching. What also can happen on this high latitude reef is that we can get extremely low temperatures at some times of the year. So this is much less common. But under those scenarios, we also saw bleaching, but less severe than at high temperatures. So this bleaching response is symptomatic of stress. You apply any sort of stress to a coral and it undergoes some sort of bleaching. But it's this high temperature that we're really concerned about. Also a marginal reef are corals up in the Solitary Islands just off Coffs Harbour. And you can see some corals dotted around. These corals don't actually form reefs like the Great Barrier Reef. The Great Barrier Reef is there because the corals secrete a lot of calcium carbonate and produce the entire reef structure. Down in the solitary islands, the corals attach to a rocky substrate and they grow from that position. Instead, what is covering most of the substrate in a lot of the solitary islands are these anemones. You can have over 300 individual anemones per square metre in the solitary islands. So they're incredibly dense in that area. And obviously you have these little Nemo fish which rely on these anemones for all sorts of things. But again, these are very sensitive to temperature change. A coral and an anemone are very closely related. If you zoom in to a single polyp, which could be a millimetre in diameter on a coral, it would look very much like an anemone. So they have very similar structures. Anemones are kind of just like big polyps of a coral, but they still have symbiotic algae living inside their tissue, so they still are at risk from changes in temperature. What we looked at was how sensitive these anemones were to temperature change. So we used Entacomia quadricolor, which is this very pretty anemone down here, to see how sensitive it was to changes in temperature. These pictures are some of the first that were captured showing how bleaching actually happens, because usually people study corals and the polyps are so tiny. But here we have an anemone which is 10-15 centimetres in diameter, so we can actually capture changes in the symbiont community. So in this picture here you can see the anemone with the oral disc in the centre, so that's not only the mouth but also the anus of the animal, so everything that goes in must come out of that same place. So here we can see a mucus ball of these symbiodinium cells, these algal cells. And eventually, in a period of about 90 seconds from this figure over to this one, we see the release of all these algae. So it's a very quick process. What we found again was that in these anemone communities, they're still living very close to their upper thermal limit. Just like coral reefs, a change of 1 degree can result in bleaching. Some work we're beginning to look into now is how important the other part of this symbiosis is. So we have the symbiosis between the animal and the algae, but there's also the fish, the anemone fish, which play a very important role. So you have usually a pair of anemone fish living within an anemone, and they reside in there for protection, but while they're in there, they also release a lot of their waste, and that's captured by these anemones. So what we are beginning to think, and there's beginning to be evidence to support this, is that fish that live inside an anemone provide the anemone with some sort of resistance to change in temperature because they have an additional food source. So an anemone which bleaches and loses its algae still has its anemone fish, and the waste from that fish can be used as a food source. But in a lot of reefs, many of these little Nemo fish are taken for the aquarium trade, so we're trying to show that by maintaining a healthy population of anemone fish, we'll also sustain a healthy population of anemones. So it's a very closely controlled symbiosis. So a lot of this work really requires us to go down to a very fine scale, so we're looking at down to the nanometers, even angstroms. So what we begin to look at is something large. We look at a coral. And then we think, okay, what's happening within the polyps of that coral? Then if we look down inside the cells, we can see these individual algal cells inside the coral. Then inside those algal cells, we can look at different components and compartments within those algae that drive photosynthesis. And that usually takes place on what we call these thylakoid membranes. So here we have a picture of one of these algae. This is taken on an electron microscope, and it's incredibly small, about 10 microns in size. So as I said earlier, that's so small that you could fit 100 of these end to end within the space of 1 millimetre. So a lot of this information, understand, is required surprise us to understand what's happening at this scale. But what we're constantly trying to do is scale up what all this means for a global ocean. So it's constantly a difficulty which we find as scientists to look at something so small but relate it to real world global problems. The next part I was going to quickly talk about is marine diseases. This is an emerging problem which we're beginning to see more commonly in lots of places around the world, and here's a few examples of diseases of corals on reefs. On a coral reef, you can imagine in your head it's incredibly clear water, it's beautifully clear aqua water. Within 1 teaspoon of that water, there is 1 million bacteria. Also within that teaspoon, there's 10 million viruses. So the microbial community within all seawater is incredibly complex and incredibly diverse and dynamic. It's constantly changing. There's always infections and pathogens attacking each other within seawater. When we go swimming, we don't really usually have to worry about this because most of those bacteria and viruses have no action on our bodies. But on coral reefs, a lot of these pathogens are becoming more of a problem. There's a whole range of different types which are of concern. The reason we think that it's becoming more of a problem is because a lot of these pathogens become not only more virulent but also more abundant during periods of a high temperature anomaly. And at the same time, the coral experiences stress, so might be becoming more sensitive to those types of diseases. So the next few slides are just some pictures which have been taken on the Great Barrier Reef of some of the most common diseases. This one is of a white band disease, and if you came back the next day, that band will probably have progressed up to about here. A day later, it will probably be up to here and up to there. In the space of a week, this entire colony could be completely dead. This one's like a coral tumour. You can see the branch coming up this way, and outside of the normal structure has grown this strange tumour. It's the only way it can really be described. Now, to understand the impacts of these diseases on corals, we can again use an instrument which looks at the health of the algae that live inside the coral. So the algae which sustain the life of this coral. Now here we have just a normal photograph, and we can see, and that's just expanded from this colony here. And we can see healthy tissue at the top. We can see a lesion border, either side of which we have an infection or no infection. And then we have an exposed skeleton. So this infection has worked its way out from the base of this colony up towards the top of the colony. Now if we look at this same colony under this instrument, we can see how it affects photosynthesis. Down here we can see a measure of photosynthetic health. So it's telling us how healthy are the algae that are living inside this coral. Interestingly, what we found was that across this lesion border, there's virtually no gradient of change. So the effect of this disease is only on the animal. It's consuming the animal tissue and working its way rapidly across the colony. So it's caused by little protozoans, so these pretty small little types of organisms which can travel across the surface and consume the coral tissue. This one is a different type of disease. It's actually by a virus. But again, it's a very similar type of response. So if we have just a photograph up the top, we can see this lesion border. On the left, we have the bleached tissue where the virus has begun to destroy the coral, and on the right, we have healthy tissue. And if we just look at this picture here, we can see that there's little dots within this area of infection. So some tissue is sensitive, some tissue isn't, but it's constantly affecting the animal. So there's beginning to be more evidence that the pathogens that are of concern in corals on the reef really attack the animals over the algae. So when it comes to changes in temperature, direct impacts go on the algae. But when it all comes to disease, which is being promoted in frequency by temperature, the effect is on the animal. So it's coming at it from both ways. Both the animal and the algae are suffering. So we're beginning to see these diseases become more common and more virulent. And the resilience of the host is decreasing, so disease is happening more often. Other things that are also related to disease are human activities like the ship ballast. When a ship comes into port, it releases a ballast which it might have taken from another port, introducing a whole new set of potential pathogens to a different environment. Okay, on to the next big section which I was hoping to concentrate on, is all about the other part of the CO2 problem. So it's not just temperature we need to worry about with carbon dioxide, it's also ocean acidification. So if we look back over the recent history in the last 250 years, we can see that CO2 concentration in the atmosphere has changed from about 280 to 396 parts per million. Now those units just mean that for every million molecules there are 396 CO2 molecules. So it doesn't sound like much, but it's causing huge changes to the chemistry of the ocean. And this change from 280 to 396 parts per million is at least 10 times, if not 100 times faster than has occurred for millions of years. So it's all about this rate of change. We may have seen a pH of the ocean as what it is today, but the rate at which it's changing is faster than we've seen for millions of years. So it's been actually 15 million years since we've seen the ocean at the pH at what it is today. What's great about the oceans though is that it's a really good sink for all this carbon dioxide. It can absorb a lot of what we release. So about a third of what we release is absorbed by the oceans. And that's a really good way to slow the effects of rising temperatures. If it wasn't for the oceans, today we'd be at 450 parts per million. So the oceans are absorbing a lot of the CO2 and slowing the process of warming. But it's changing the chemistry. How does it do this? Well, it forms a weak acid called carbonic acid when CO2 dissolves. When it dissolves, it changes pH, and pH is a measure of acidity. Today, the ocean pH is 8.1. 250 years ago, it was 8.2. 0.1 change doesn't sound like much, do we really need to worry about that? The way pH is measured is on a logarithmic scale, so that 0.1 change is an enormous change in the acidity, and that's measured by the concentration of hydrogen ions. So just that 0.1 change means that there today are 30% more hydrogen ions in the surface ocean than there were 250 years ago. Thinking about the size of the ocean, that is a huge change. This picture down here demonstrates the geographical variation in changes in pH and how that can be influenced by local processes. So in some regions, especially around the polar regions, we're seeing the greatest change in pH. The reason for that is because it's a lot colder. In colder water, the absorption of CO2 is much greater. So we see faster changes of ocean acidification. If we continue as business as usual in our release of carbon dioxide, we could expect by the end of this century a further drop by 0.3 or 0.4 pH units, which is enormous. So we're seeing a reduction in ocean pH because of all this CO2 in the atmosphere. The other part of the problem, instead of just changing pH, it's altering this molecule. This is called carbonate. More CO2 equals less of this carbonate molecule. If you think about a coral, it forms this skeleton, and that skeleton is made of calcium carbonate. Our bones are made of calcium phosphate, so it's very similar. A coral is very similar to our bones. Corals are actually beginning to be used as bone grafts in some people because it's so similar. But because we're seeing this change in ocean pH, we're seeing less of this carbonate ion. So the ability for this coral to produce its calcium carbonate skeleton is becoming much more difficult. But it's not just corals. There's all sorts of things in the ocean which produce calcium carbonate skeletons. There's all sorts of animals, many of which we love to eat, but there's also things like these single-celled protists and lots of marine plants. So there's lots that we need to be concerned about when it comes to ocean acidification. It's not just going to affect some pretty animal in the ocean, it's going to affect the economy and what people eat and all sorts of things. So it is something that people should be concerned about. This is a bit of a projection into the future. So here we are today, well, a few years ago, 2005, and here we are towards the end of the century. And this is a map of how easy it will be for a coral or a clam or a prawn to produce its skeleton. In the blue, that's good. That's what we want to see. That's where an organism can easily produce its calcium carbonate skeleton. As we go towards the oranges and reds, that's where we begin to be concerned. The ability for these organisms to produce that skeleton becomes very marginal. As we get closer to 1 in this scale down here, so the closer to red, there's a greater chance of dissolution. So if a coral is producing calcium carbonate to make its skeleton, under CO2, high CO2 conditions, that can be reversed, so it can begin to dissolve. So we can see entire coral reefs beginning to dissolve in extreme circumstances. So obviously this is going to affect how fast something can grow. It could lead to something dissolving. And in such a case, if something is very vulnerable to predation, then it needs to be able to defend itself. If it doesn't have that same strength of a skeleton, then it's going to be more susceptible to predation. Coral reefs around the world are great for many reasons. One of them is because they provide coastal protection from things like tsunamis. For the coral reefs to be there in a healthy condition, they can absorb a lot of that wave energy. But if they become destabilised from ocean acidification, their ability to withstand wave energy will be reduced, so they can be removed or broken by wave energy. Eventually we could begin to see loss of coral reefs and definitely altered ecosystems. So the dynamics of these ecosystems and what eats what and what's available for consumption will dramatically change. Now this is just a little example I've put in. Again, it's back to these single-celled algae, which are really small. That's about 10 microns from side to side. And they're called coccolithophorids. They look pretty cool under an electron microscope, and these little circles which produce their skeleton are again made of calcium carbonate. This is a satellite photo taken in the English Channel, and this big white bloom is because of billions or trillions of these single-celled Amelia huxleyi coccolithophorid algae. So there's lots of them. So what are they doing? Well, it's because of them that you have the White Cliffs of Dover. They are produced because of these tiny single-celled algae. So they make huge changes to the landscape. They're incredibly important. But what some people have done is to incubate these algae under different climate change scenarios. So if we change the CO2 concentration from what it is today to a higher CO2 concentration, something we can expect in the next 50 to 100 years, we begin to see changes in how the calcium carbonate is deposited in this species. So we see malformations and reduced calcification, changes that obviously indicate some sort of damage to the calcification process. On a coral reef, obviously there's corals, but there's a lot of other things living there. They are some of the most biodiverse ecosystems in the world. And these are 2 examples I've chosen to demonstrate that it's not just corals that we have to worry about on a coral reef. This is called Halimeda. It's a single-celled algae. It can get to 15 centimetres tall, and it has a deep root structure. Single cell. Very cool, but it produces this calcium carbonate skeleton. This one is a single-celled protist. It can get about 1 centimetre in diameter. It's called a foraminifera. And we've done some work to try and understand how sensitive these guys are. We've looked at a whole bunch of different measurements to see how sensitive they are to a change in temperature and CO2. We have to look at these two things, both CO2 and temperature, in combination and in isolation to see which one has the most effect, but together does it have even greater effect. These are some photos we took under an electron microscope, so we're at a very fine scale. This little scale bar here is 1 micron, very small, and what you're looking at are individual calcium carbonate crystals. On the left-hand column, we have 2 species of that green algae I showed you, and down here we have that single-celled protist, the foraminifera. And on this side, this is the control condition. So present day, pH 8.1, 28 degrees, pretty happy on a coral reef. This one is after 5 weeks at extreme CO2 conditions, so something we'll expect towards the end of this century, and high temperatures. And we found something we didn't actually expect. First of all, under ocean acidification and warming conditions, we saw thinner calcium carbonate crystals. Okay, that makes sense. They can't produce as much calcium carbonate. What we did see was that there are actually more of them. More of these crystals. Smaller, but more. So it was telling us that the process of calcification, the initiation of crystal formation, and the termination of crystal formation is happening much faster. So it's affecting the physiology of these animals and how they produce their skeletons. So why do we care about this? Well, if we think about a coral reef structure, this big three-dimensional complex coral reef, towards the edges of the reef and under storm conditions in the centre of any reef, you can have a lot of wave action. There's also lots of herbivores trying to remove this type of algae from the reef structure to consume it. Calcium carbonate is a great way of avoiding being eaten. It's not very tasty, it's hard, it's going to chip your tooth if you try and eat it. A lot of fish try. What we're beginning to see though is that some of these measures of biomechanics, how strong these algae are, how they can resist consumption or breakage from waves, that's beginning to decline. So under future climate change conditions where we expect to see not just higher temperatures and more CO2 in the ocean, we're also going to expect more intense storms. So the removal of structures such as a halimeda plant will be much more common. These are just going to be a few interesting photos taken from a recent study back in 2008 on a very interesting ecosystem in Italy. What they discovered was these CO2 vents coming out of the bottom of the ocean. And these vents are neighbouring relatively healthy and what we would call normal ecosystems. And there's a gradient of pH change between the areas where all this CO2 is bubbling out towards an area where there is no CO2. So it's like a natural mesocosm to understand what's going to happen into the future. And what people have done is performed a series of transplantation experiments where you take an organism from one place to another, or you can do measurements and monitor what's there and see their condition. These sea stars, they actually have an internal skeleton made of calcium carbonate. But if they're in a high CO2 environment, they are 80% lighter because they do not have the ability to produce their skeleton. Also absent in areas of high CO2 were things like these snails. They have their shell which is made of calcium carbonate. This is a seagrass blade covered in this pink type of algae. That was absent. Things like abalone, they were no longer there. They couldn't produce their skeletons. If we look at the larval stage of some of these organisms, so in a sea urchin, they don't look anything like their adults, but this is a juvenile sea urchin larva. Under normal conditions, they look like these sort of satellite structures, but again, you incubate them under high CO2 scenarios and their growth and morphology greatly changes. So if we look into the future, what effects might we see and how far into the future will those effects occur? If here we are today around 2010 in the middle here, and in the future we could expect to see impacts to cold water corals, deep corals which don't actually harvest light, they only consume prey that goes past their tentacles. They're going to be really vulnerable to start with. Then brittle stars. We can see impacts to some of the snails and mussels. Further up, oysters are going to experience reduced growth. Sea urchin embryos and larvae, the developmental processes become damaged. So these are some of the future effects we can begin to see, and these are plotted on a business as usual scenario. Hopefully we can take one of these pathways instead and not experience or begin to observe such changes. Alright, just on to the last few slides now. I just wanted to talk about some of the work I've done in Antarctica. Unfortunately I didn't get to work too much with these beautiful penguins, but instead we worked a lot on these tiny little algae. But these algae are so important. They live on the bottom of the sea ice, and the krill will consume these algae, and then the penguins will consume the krill, and then the whales consume everything else. So these little algae are there, and they sustain all the life that we know it under the sea ice in Antarctica. And we call them extremophiles because they're living in such an extreme environment. The temperature in this ice core can get to -20 at about 5 to 10 centimetres up from the bottom of the sea. So they're incredibly cold. They form these little brine channels which become very saline. They can be 3 times more salty than normal seawater. Living inside ice, there's not much capacity for gas exchange. So how do you get oxygen in for respiration, and how do you get waste out? So they're living in a very extreme environment, but they have maintained very high productivity, and they sustain all life down in Antarctica under the sea ice. What we're beginning to see though in Antarctica is that the sea ice is thinning, and the amount of snow on top of the sea ice is also thinning. This means that more light is penetrating the sea ice. Obviously light is important for photosynthesis in these sea ice algae, but there is such thing as too much light, and too much light can kill any type of photosynthetic organism. So this is the type of algae you might see when you look under the microscope. They're really beautiful. They're called diatoms, and they have glass cell walls made of silicon oxide. And you can get all sorts of shapes and sizes, and yeah, they're incredibly beautiful. So when we worked with these algae, we took cores about 1 metre deep under the ice, and we cut off the end bit, which has a nice bit of algae in it. And back on the ship that we took down to Antarctica, the Aurora Australis, we were able to incubate them under conditions which we could manipulate. Now this figure here shows changes over time in hours, up to 5 hours, and this is just a measure of how healthy these algae are, how well they can photosynthesize. In most places about 1 metre under the ice, they'd experience very low light intensities. So on the midday in Sydney, if it's not cloudy, you'd expect there to be a light intensity of 2,000 units. Under the ice, they usually get 10 to 50, so very low, but it's enough to sustain photosynthesis. What we did was double that, and then also quadruple that to see how that would affect their ability to photosynthesize. And these are light intensities so high that they probably have never experienced it. They may in the future, but at the moment, they would never have experienced something so high. And what we found was they were incredibly plastic. They were very flexible. They could bounce back from any sort of light intensity which we threw at them. These algae are living already in such an extreme environment, but they can take yet another stress and deal with it with no problem. So this is a bit of a good news story, and something which we weren't expecting, but these algae could come back after a period of recovery and show that they were just as healthy as when we started. So what's the next step in some of this research? Well, now, concentrating in the Sydney region, we want to know what's happening right on our doorstep. And living in Sydney, we're actually in a climate change hotspot.